Good morning, everybody. On behalf of the panelists and our co-organizers, I'm delighted to invite you to the Spring European Aquaculture Technology and Innovation Platform Forum. My name's David Baset. I'm the General Secretary of EATiP, but we're delighted to be running this event with our fellow European technology platform FABRETP and the European Forum of Farm Animal Breeders. And I'd like to, right at the outset, extend my thanks to the support given by a variety of colleagues at both those organizations. As a European technology platform, it's our purpose to take a multi-stakeholder approach to promoting aquaculture research and innovation. And we're delighted that the registrations for this event, at least from what we can see at this end, bear out a cross-multi-sectoral interest in this particular topic. Our online thematic forums were born out of a lockdown necessity to communicate with our membership, but they've proved very popular and very successful with a growing attendance, and we're delighted to continue the series. We've covered a variety of themes so far, but today we're turning to a more technical and scientific area, gene editing in aquaculture, which is perhaps one of the areas demonstrating the most rapid advances and aspects of innovation. And I'm sure that many of us are eager to learn much more about. Although, having said that, whilst noting it's technical and scientific, gene editing is in no way rarefied. And as we're going to be shown this morning by a variety of talks, the application of these techniques, it's broad and relevant to many key aspects of our sector, from aquatic animal health and welfare to environmental interaction, human health and food safety. There'll be a series of talks, some polling and some questions, and an opportunity for discussion at the end of the event. But you've not registered to listen to me, so I'm delighted to pass on to our co-host, Anna at EFFAB, who will introduce the platform, her platforms, and present the housekeeping rules for the event. Over to you, Anna.
Thanks, David, and good morning, everyone. So I'm Anna Granados from FABRETP. We are hosting, I'm not going to repeat everything David said because it's quite similar, but yeah, we are happy to welcome you and to organize this event in collaboration with the European Aquaculture Technology Platform. We collaborate in other manners the rest of the year with them, and that's why we are happy to host this webinar. That also this series of webinars of FABRETP and EFFAB started with the COVID pandemic, and but we are aiming to continue during 2022, of course, with less webinars than we had in 2021, but engaging with topics related to animal genetics and probably with other collaborations in the future. We are hosting today genome editing in aquaculture, because aquaculture is a very important part of our membership, and the genome editing research is, I will not say more well advanced in aquaculture than in other species, terrestrial farm species, but there are very interesting applications, potential applications that we would like to tackle today. And with this, I will share the screen with the agenda to remind everyone who is talking. Just a second, I have to share my screen in presentation mode. So just to remind you, you are not here to listen David and me, as David said, but we had an opening talk from the Commission, and Diego will explain very general things about genome editing, and we will go through more specific applications and research after with Anna, Ross, which is we have a video, Tone-Kari, and Alan Tinch. We had a panel discussions after, so I'm going through the house rules now. Sorry, if you have questions, you can introduce them in the QA box, please. That's the best way to interact with you. Some of the time the speakers will probably answer directly there. And we will, it is not the case, we will take your questions for the panel discussions. We are aiming to have a long panel discussion. If you have any technical issues, you have the EFFAB email address or the Norali one. So Norali is in the chat. She had put what you can ask her. You can ask her if you have any problem. Don't forget that the webinar will be recorded and shared in our website and social media and as the European Aquaculture Platform too. And you will receive a short report after in a few days or weeks, because we have our annual general meeting next week. So with this, I give the floor again to David, please.
It's my pleasure to introduce our introductory talk at this point in the event. We normally set the context with a policy status of what we're looking at. And I'm delighted to introduce Frank Swartenbroux from the biotechnology unit of DG Santé at the European Commission. Frank originally graduated as a vet from the University of Ghent, before doing some more specialized work looking at hygiene of food of animal origin, prior to moving on to working for the Belgian competent authorities. From there, he moved on to working at more cross-EU level, joining the European Commission in 2007, where he started to work at DG Santé on residues in veterinary medicinal products and contaminants, before moving into the biotechnology unit. Frank, thank you very much for making your time available to us this morning. And we look forward to being guided through a policy status to set and reference our talks this morning. Thank you.
Thank you, David. I guess you can all hear me because somebody helped me by unmuting from distance. That's already very nice.
Absolutely.
So I would like to thank you first for inviting me. I always like with being here and learning a lot, where I then most of the time end up giving the most boring presentation, but do learn a lot from the presentations that the other participants are giving. But I guess it is true that we need to keep you updated of where we are with the policy on gene editing and or new genomic techniques or whatever. New breeding techniques or whatever they are called. There's a variety of names. We kind of try to stick within the Commission to new genomic techniques, as this was the term that was kind of inserted by the Council when they asked us to do a study. So I'll just give you a short update of where we are with the policy initiative. I think I don't need to remind you that how do I go to the next slide? Yes. So to remind you about what happened before, we had the court ruling. We had kind of a dramatic situation where everybody didn't know or nobody knew exactly what to do. And we were asked by the Council to make a study about the impact of the court ruling on genomic techniques. And the outcome of this study was the Commission considered that we would need to take a policy initiative on certain aspects, not on everything, but to start at least with one part. And this was then later confirmed by the Council of Agriculture. The ministers said that it was a good idea and that they wanted us to move forward. And also the European Parliament said that this would be a good start. So this is where we started with the policy initiative, maybe to situate it. You know, all these things which the Commission is working on in the European Green Deal, going from eliminating pollution, etc., protecting nature, going everywhere, doing farm to fork. And we are, with our policy initiative, are in the farm to fork aspect chapter, I would say. The overall goals are very clear of the farm to fork strategy: reduce the environmental and climate footprint of the food system. There is, of course, a lot of people who are worried about the way we are in, not only in Europe, but in big parts of the world producing food, and whether this is really in the long run sustainable if we need to feed 9 billion, 10 billion, 11 billion people, how this would be possible. And that we also need to create a global transition, so not only us, but also other words, to competitive sustainability. I mean, it needs to be sustainable, but it also needs to be a little bit competitive. It needs to be good prices and good prices also for farmers, because I think in case you aquaculture breeders, because if there is no income, I don't believe it will be very sustainable. People will give up and do something else. But we also have to use the new opportunities that are there and create a robust and resilient food system. That's clear. We are a bit worried. I think also now in times with the Ukrainian situation and the war, we do see that the system is not maybe as robust and resilient as we had all thought one year ago. That we do need to realize clearly that this is that we are somewhere a little bit vulnerable. So what did the Commission studies say? Well, we concluded that our actually GMO legislation and risk assessment is not very fit for purpose for NGTs. It needs an adaptation to the scientific progress, technological progress, because otherwise this cannot be really promoting NGTs and their products. The current oversight we have and the risk assessment requirements, they are not tailored to the diverse risk profiles, which is not really, I would say, as a surprise, because the legislation is almost 20 years old and was kind of tailor-made for the situation 20 years ago. And it's clear the situation has clearly rapidly evolved over the last 5 to 10 years. We also think that, of course, NGTs can clearly contribute to the Green Deal and the Farm to Fork objectives as to the more competitive economy. This being said, we also are not, I would say, preaching that NGTs are the silver bullet to solve all problems in Green Deal and Farm to Fork objectives. For us, NGTs, gene editing and all these techniques are an important tool, but only a tool. Other tools are there also, and I think everybody, I mean, with the ambitious goals for Green Deal and Farm to Fork, everybody working on things should do their utmost best, because the individual chapters will not do the trick in the long run. I mean, everybody will have to contribute with his responsibilities to reach the ambitious goals by 2030 and 2050. It's not. Some of these goals are very close by. So there have been concerns that were addressed, and we should express that we should address. So the application should not undermine the other aspects. And of course, very sensitive. It's very clear in the Green Deal, the European Parliament and also the governments and Council clearly wants not to undermine the organic agriculture. That is, whether it's organic, I would say, agriculture or organic aquaculture, it is, of course, an important part of the future. So what is the initiative? The first part we would say the most urgent we considered was to make a legislation for plants produced by targeted mutagenesis or by cisgenesis. This is what we said we would start with, because I mean, we do think that plants were a bit more advanced looking at the NGT studies, more products were close to market. I have to admit that the study is more than a year old now. The pace is really fast, and I do see that in aquaculture things are moving also extremely fast, and that well, I'm a bit surprised by the speed everything goes, and I also know the speed of which we can make legislation, drafting it, and all the obligations we have to do, and at the time it takes. And I'm a bit worried sometimes by the speed of the progress and the thoroughness, not to say the lack of speed of our proposals, where we need to go to Council and Parliament, of course, also. So what is the scope and the objectives? We do want to create for these plants a proportionate regulatory oversight. It's clear we want to maintain a high level of human protection, animal health protection, and of the environment. We're not going to give in on a number of the goals that have been set over the last years. I also think that citizens would not accept that we would reduce the environmental protection at this moment, nor would they accept that we lower the standards for human or animal health or even animal welfare for that part. We want to enable safe plants to provide benefits and contribute. Okay, that is very clear, but also to the biodiversity factors. And we do want to enhance the competitiveness of the EU, and we ensure the good functioning of the internal market. Especially the last points are getting a bit more important, I would say, the last months. So what do we want to considering these things, we want to look at the risk assessment and the approval requirements. They are extremely, I would say, prescriptive. Some, I would say, economic blocks outside of the EU would say that they are too prescriptive and too strict. But we want them to be a bit more proportionate to the risk profiles. And we want to move a little bit more to a case-by-case basis rather than just making one blanket legislation that covers everything. There's a big request, a big pressure to include the sustainability analysis, to examine whether and in which ways these products really contribute to sustainability. I guess you know from the GMOs, this is the eternal debate. People just say, is it really sustainable with all these pesticides resistance, herbicide resistance crops we have, when we, on the one hand, we authorize these crops for growing, not so much in the EU, I have to admit, but on the other hand, we keep on saying that we're going to reduce the pesticides used by 50% or 30%, etc. So this is a thing that is, we feel a lot of pressure from everywhere. We also want to have to look into traceability and labeling, because it is also very clear that contrary to other parts of the world, the European consumer is very keen on knowing what is on his plate. Labeling and traceability are extremely important. I think it has probably to do with the subsequent, in the 90s, BSC and dioxin crisis, and a few very big crises we've had in the past, that the European consumer is very much critical about all this. So it's clear we will have to address these provisions also for NGT products. And we need a mechanism to be able to rapidly adjust the elements of the legislation, because that is something that is very clear. The older legislation, the old framework legislation, the directives and the GM food feed regulation, they are a bit, it's a bit difficult for the Commission, I would say, to adapt them to progress. I mean, we have a very heavy mechanism on how we have to do this. There's a lot of procedures we need to follow. This is all considered as being changes to the basis of the act, which means we cannot do this through an implementing regulation at Commission level, so voting simply in a standing committee. These are things we need to get through the Council and Parliament, and that then, of course, results in the heavy procedures with Inception, impact assessments, etc., which take an awful lot of time. So, which are the impacts we check? The economic impact, of course, that is very important, because, as I said, if it's not economically sustainable, nobody will want to invest any money in this. The fundamental rights of the citizen, the social perception, which is very active. The social dimension is very active. I would say even certain, not really like the one on the right, fundamental rights. It's not always, in my feeling, a balanced social interaction. Not everybody is speaking as loud or not representing the same size of groups. Or some people are, some smaller groups are sometimes, I feel, shouting, while some bigger groups are more on the sideline and watching what's going on. And you can wonder who is right and who's wrong. We also, of course, want to do the simplification of the administrative burden. It needs to be, I mean, that is becoming annoying for everybody. It's already since many years that we try to do it, and each suggestion where it can be done is welcome. And we need to look at the environmental impact as well. We try to do consultations and outreach. We've done the high-level event on new genomic techniques already in November last year. It was very successful. A lot of people participated. I think the recordings are still online available. It's only 1 year, so not even. So normally they are still online. You can still check this and see what was said. It was, for me, it was a very interesting and a very interesting day. And then we had the second step after that. We did the Inception impact assessment, which used to have a nicer name, the roadmap, which was open for feedback for 4 weeks until October 2020. We received over 70,000 contributions. That was a lot to analyze. Thankfully, we had help from the GSC, who seemingly have data mining machines and text mining machines who can help us out, because Not all of them, you should understand, are individual contributions. They were individual contributions from citizens or stakeholders, but with the text mining, we could discover that a large number of them are actually coming from the same origin and are actually concerted actions to react on the roadmap. In total, we received contributions from 91 countries, which is quite a lot if you include, okay, even if you discount the 27 member states, still 64 non-EU countries who went through the pain to reply to us and give their views on how this would influence the position and the trade amongst others between them and us, and how it would what they think we should do. Sometimes a bit paternalistic, but sometimes also well meant, I would say. So the main topics we discussed with our correspondents in the reactions were the initiative, the impact, Inception impacts, and the GMO legislation, the risk assessment, sustainability, traceability. Some of these comments were really technical. I mean, it depends on which group they are. There are, of course, also comments which just say, leave it as it is, the GMO legislation, I don't want you to change anything, I want it to stay as it is. There was complaints about information, well, concerns about information to consumers and operators, and also the liability and the cost of a possible contamination if things went wrong, which is, of course, this bullet is much linked to softer regulation and a fear of a number of people that we would go down the line like you have in some other countries, like for a complete deregulation, that you say you don't need to register, you don't need to get an authorization, you can just bring it on the market, and then if something would go wrong, it would become an environmental disaster. Who is going to pay for this? Last part, of course, is the intellectual property right, which is not new and which is not limited to the NGTs, which is not even limited to the GMO discussion, which is broader. Intellectual properties plays a more and more important role. There is an economic model of earning money through the intellectual property rather than through the product itself, and this is considered as worrying by a number of an increasing growing group of people that think that this is going to limit the access and the choice of the people who actually produce the food. So where are we in the timeline? Because it's an update of where we are. You can see there the initiative we announced in April 2021. We made the Inception impact assessment by my colleague Petros, mainly produced by 20 September. In November, we had a high-level event. In 22, we are going to do the impact assessment. We are currently working on it. We are almost there. We have to still work a little bit, but okay. The impact assessment will be open in the second quarter of 2022 for an open public consultation. Again, I know we are already in the second quarter. People tell me, I think I received calls the very first days of April where it was. And I do need to remind people that the second quarter does run until the 30th of June. So we are not yet in June, but we are still on time, I would say. After that, we will give people time to give the public consultation and look at it ourselves. That will be during our summer break, and then we will do the actual work and say, now, what is the possible legal proposal we are going to make? How will it actually look like? What will we draft? And then we will put it on the table in 2023 for discussion in European Parliament and Council. And then we will see what is the result. We hope, of course, that it will be a favorable result, which satisfies everybody, which I know is not always possible. But okay, that is how it is. So that would be it for me. I'm available for questions if needed. And I will now click stop share and see whether it works. Voilà.
Thank you. Thank you very much. Thank you very much, Frank. Thank you very much for this insightful presentation with the last news from the policy actions from the European Commission. We have questions for the panelists at the panel discussion, so you can post them in the QA box. We are going to launch just 2 polls now to ask you about your background. We will have other questions on Mentimeter, as you have seen before in the first presentation. So we would like to know who you are. But it's all okay. So 50% of research institute, that's not very surprising. Also from companies, 34%, 6% from NGOs, and 3% of policymakers, and other we don't know. So the second question is just down. What is your motivation to join the Forum on Genome Editing in Aquaculture? Well, I'm not directly involved, but having an interest in this field. But the second one is I'm currently involved in work, and that's very good that scientists involved in this field have joined in. So stop sharing and let's go to the next speaker, who is Diego Robledo. I suppose you are happy we can pronounce the R, sorry. So Diego is a scientist, is a Spanish scientist, having a PhD, graduated and having a PhD in the University of Santiago de Compostela, but he's working in aquaculture genetics and genomics at the Roslin Institute in Scotland. And the main interest of his research group is to understand responses to pathogens in aquaculture species, and especially differences in disease resistance, both within population and between species. So he's going to give us an overview on applications of genome editing in aquaculture. And yeah, Diego, are you mute? No, well, you can hear you, and I suppose you are going to start your presentation. So I stop here and give you the floor.
Thank you very much, Anna, for the introduction, for pronouncing my surname properly. That's appreciated. And thank you very much to you and to EFFAB and FABRETP for the invitation. As you mentioned, I'm going to give a general overview about genome editing. So I'm going to basically set the scene for the next speakers who will get a bit more into the topic. Let me just share this. So you should be seeing the presenter mode at the moment, I think. Is that correct?
We can see the next slide too. So you have to hide.
I have to hide this.
You have the three dots.
Yeah.
And you have to hide. Yes, perfect. Now it's perfect, Diego. Thank you. Yeah, go ahead.
Perfect. So yeah, as I was saying, I'm going to set the scene basically and talk a bit about genome editing and what it is. So what is genome editing about? Well, the definition of genome editing is targeted changes to the DNA of a cell, cells, individuals. And I think that targeted is key here. We target the region of the DNA of our genomes and modify it. What does it mean by modifying the genome or the DNA? Well, the DNA is formed by over 500 million pieces, depends on the species, between 500 million and 3 billion, more or less. And these pieces are what we call nucleotides, and they are combinations of 4 different nucleotides. The DNA is a combination of 4 different nucleotides, which are abbreviated with 4 letters, which are RCTG. So here you can see genome editing in this picture. For example, we'll be changing a thymine by a C cytosine. But it could also be removing a small piece of the DNA, and it could also be adding a small piece of DNA. So all this happens naturally. Genome editing, the modification of the DNA, not targeted, but random changes in the DNA occur naturally. Okay, and they are in all of us. When we start, we are a single cell organism, single cell embryo, but as that single cell multiplies to develop the whole organism, all our tissues or our cell types, changes occur. Just by mistakes in the replication of the DNA, changes occur. So we at the end, all of us are a mosaic of different types, of different DNAs. Tiny small changes, but changes after all. And if those changes occur in our germ cells, in our gametes, they can be transferred to the next generation. So we could say in a way that the DNA of our children has been edited in the natural process of life. And these changes are fundamental, are the substrate of evolution and selection. these changes that occur in our DNA and happen from one generation to the next, will have given rise to the whole diversity in animal life, in life in general, and also are what we use to select animals. These changes can affect traits of interest, and that's what we select in selective breeding. Those changes in the DNA that can produce variation in the traits of interest. So nothing strange. Changes in the DNA happen all the time. And what we do use today for genome editing in the lab is CRISPR-Cas system, which I'm sure you have all heard about. So before I get into how they work and what they actually do, I want to touch a bit about the origin, because probably many people will know that these are the CRISPR-Cas systems are an immune system of the bacteria that was discovered by Francisco Mojica from the University of Alicante. And they are actually present in most bacteria, around 50% of the bacteria species, including E. coli. So probably around you today, there are bacteria that have the CRISPR-Cas system. They are just the defense mechanism that the bacteria use against viruses. And how they work is that they are natural DNA scissors. So basically what they do is they cut the DNA of viruses that get into the bacterial cells. How does the process work? Well, when the virus attaches to a bacterial cell, they introduce the viral DNA into the cell. Part of this viral DNA is copied into the genome of the bacteria. This is what is actually the CRISPR part of the system, where you have different fragments of different virus with different colors, and then just repeats between them. And this fragment that comes from the virus that infected the bacteria is expressed into what is called a guide RNA. The guide RNA basically directs the Cas complex, the proteins that are the ones that actually produce cuts in the DNA. So the guide directs this Cas complex to the viral genome, cuts it, and avoids the infection. And as I said, this is an immune mechanism that bacteria have all around us, and it's natural and present everywhere. But the brilliancy of CRISPR-Cas is that you can exploit it to basically edit the genomes of the species you are interested in. This is something that was discovered by Jennifer Doudna and Emmanuel Charpentier, and that gave, well, as a result of that, it resulted in the Nobel Prize in 2020, as I'm sure you have all heard about. So what they did is basically repurposing the system to target the DNA sequences or the genes that they wanted to. And how do they do that? Well, when you have a one-cell embryo, let's talk about one-cell embryo, when the two gametes, the sperm and eggs merge, have the one-cell embryo, you can insert in that embryo the Cas9 enzyme, the protein, basically, and a gRNA with a target-specific sequence that is targeting a gene of interest. And what happens is that inside that cell, basically, the gRNA will, with your target-specific sequence, will bring the Cas9 to the DNA of the cell, and then the Cas9 will produce a cut. Okay, and that cut, because the eukaryotic organisms have very good, unlike viruses, the eukaryotic organisms have very good repair mechanisms. So the same machinery of the cell will repair the DNA, and what will happen is that it's not perfect, so it will usually not repair it as it was. So for example, after a cut, what will happen frequently is that there will be deletions, small parts will be missing. We're talking about 1 or 2 pieces in a 3 billion puzzle. And what can also happen is that you can also introduce into the cell another small piece of DNA that is that basically tells the DNA of the cell how to repair it. And that allows you to produce targeted changes in the DNA. So basically, change one piece for another. That's how you would do it, by introducing another small template of DNA into the cell. Importantly, I think the Cas9, the guide RNA, the donor DNA, if you introduce those, so they are degraded within hours. So they disappear from the cells soon. They are, and all that remains are the changes that you've made in the DNA. Yeah. So what's the potential? Well, the potential is basically almost unlimited within biological limits, obviously. There's the ability to improve almost any trait. And what's important is that it's not limited to existing genetic variation. So in a way, rather than waiting for changes to occur naturally, for changes to occur over many generations, that we can exploit, what we do is produce those changes ourselves, speeding up the mechanism of improving the traits of interest. So for example, one classical, here I have 3 different examples in different organisms. One of them is hornless cattle. So this is something that actually occurs naturally. There are cattle that don't have horns. We know the gene, but with genome editing, you can easily transfer that to other breeds much faster and much straightforward than if you do by conventional breeding. And that leads to, well, improved welfare of the animals. They don't harm themselves, they don't harm humans, but also they don't have to go through the horning, which is quite a stressful and dramatic event in the life of the cattle. Here I have one from the Roslin Institute, that was done here in our research institute. So this is for resistant to porcine reproductive and respiratory syndrome virus. So by editing a small piece of the receptor of this virus, researchers here were able to make the animals fully resistant to disease, so meaning that they don't suffer from problems, from discomfort, death, etc. And the final example, one aquaculture that I think our the other panelists won't talk about today, is the fast-growing insebrine that has just been approved in Japan. And what this basically does is the food efficiency, the food conversion rate, food to growth conversion ratio of these animals is better, is 20% better. So that means they are more efficient in converting food to growth. So all of these have obvious benefits. First, clearly for food security, but also for animal welfare, also for the environment, and it could have also benefits for human health in other scenarios. So, but I've presented the good parts. Let me go also through the challenges. Well, the first and the biggest challenge, at least for scientists, is to know what to edit. You can think about our genomes, our DNA, about big puzzles with between 500 million and 3 billion pieces. And even if you focus just on the genes, which you could say are the most important functional part of the genome, there are between 20,000 and 25,000 genes in each species, so quite a lot to investigate. There are projects now funded by the European Union, such as the AquaFund, which tries to annotate the genomes of 6 aquaculture species in Europe. And what they are trying is precisely to find which species to edit. How, if we understand the genome, if we understand what the genes are doing, it's easier to know what we have to edit. So this is the main area of research, probably, where we focus our efforts. But there are other challenges, I guess, that you could think about when talking about genome editing. So one of them is the potential of targets. Biological systems are never perfect. You have a guide RNA that tries to target the region of the genome, but by mistake, you could end up targeting other regions also that you didn't intend to, but just, biological mechanisms are never perfect. Nonetheless, this can be assessed and controlled. It's not that huge problem because we can easily sequence the DNA and actually find the soft targets if there are any. And actually, the methods are being refined constantly for genome editing, reducing this problem. So it's not a huge problem. It's just something to be aware of. If you ask me about what's the biggest, the second biggest, I would say, issue before we can actually use genome editing on farm system, in our farm animals, especially in aquaculture, is their interactions with wild populations and the concerns, and that we need to consider that in terms of genetic diversity. So we cannot let, I think we shouldn't let our genome edited animals mix with the wild populations, basically. Nonetheless, that probably requires quite a lot of innovation in the sense that those animals probably in most cases will need to be sterile, and at the same time you still want to produce more generations of those animals. So there is a conflict there, and that will require some technological innovation. I know some research groups are working on this, and I think maybe Anna will talk about it later. Another possibility, obviously, is to use closed systems and just not allow don't leave any chance for the editing animals to get into the wild habitats. I say this is probably the second most important problem before we can apply genome editing. I think the most important is actually public opinion and public perception. That's the main thing that is holding back genome editing, in my opinion. So I think it's really important that we consider how we communicate genome editing when we are talking to the public. That we, all of us working on on this topic really get involved in this. And I think there are some key messages that are, it's better to focus on the positive messages, basically. I think the potential is enormous, and it can have major benefits to everyone worldwide. So it can feed the world, it can help the availability and the affordability of food. Everyone, I know it has, many people check the labels, but most people check the price, and that if genomating can make food more cheaper, well, that's a win for everyone. It can also improve animal welfare, mainly disease resistance. If you think about disease resistance, it's a huge problem in our culture, but also other traits like the horning in hornless in cattle. It can also reduce environmental impact, for example, making the animals more efficient in their food conversion, but also reducing the treatments for diseases. And it can also improve human health, first through reducing antibiotic use, but I guess in the future we could also help improve nutritional profile of our animals if we wanted to. There are some people will argument against genomating, even despite the key positive message. I think probably the things that we should say to offend is that the small changes that occur, they occur naturally. They can occur naturally. They are naturally present in the population many times, and it's not a different process than that happens through evolution all the time. Right? I have two pictures here I like to present when I teach genomating in the university. These two pictures differ in 1 out of 2,000 pixels. So I basically changed 1 pixel in one of these pictures. I cannot find it. I don't remember which one I changed. I know it's red, but I don't remember. I cannot find it. So in genomating, you basically change more or less 1 in a billion pieces. So it's really a tiny change compared to the whole genome of the species. And then the second counter argument, I think they will argue that wild populations, people will argue that, well, rightly will argue that wild populations need to be protected. And I agree with that, but I think we can protect the wild population from those impacts. And that's what legislation is for. I don't want to say much more. I just want to end up with pictures of animals that are genome edited, some of them, and some of them are not genome edited. You probably won't be able to distinguish them. So they are basically, they look the same, they behave the same, they have the same properties, they're all happy, and yeah, all good and beneficial for them when they are edited. So nothing else. Thank you. If anyone wants to chat, like ask any questions now, otherwise you can ask me by email or Twitter. Happy to reply anytime. Yeah. Thank you. We'll stop sharing.
Thank you, Diego, very much. I do not have a technical background in the subject at all, so I very much appreciated your talk. I have a natural bias. I was brought up just outside Edinburgh. So the University of Edinburgh, the Dick Vet School and the Roslin Institute are all names I'm familiar with from growing up. And thank you for a very clear and easily accessible introduction to the topic. I think if nothing more, then we need to be prescient of the importance of communicating many of the aspects of the subject area, given the sensitivities that can surround it, and you've done that very clearly and succinctly. We're now going to move on to do some slight polling. We've had some general introductions to the policy contents and to the subject area of gene editing, thanks to Frank and Diego. My colleague Catherine has just posted a link to a Menti poll, which you should be able to access. And Catherine will introduce the questions that we're asking you. The Menti poll, I'm sure you'll all be familiar with this by now. You go to www.menti.com and the code number that you have to enter is there. It appears on the slide just in front of you, and the details are in the chat. So while you all scramble around for your telephones or open up the other screen on your desktops, depending how you're operating, you can bring the questions up. You're all getting there. I begin to see the people registering now. So we'll just hang on a couple more moments whilst people find the poll. There's a lot more than 7 of you, so hopefully you can all quickly register. And I believe Catherine is going to introduce the questions.
Okay, so
thank you, Catherine.
Here is the first question. What target traits would you consider to be of the highest priority for aquaculture in commercially applied gene editing? You have 5 options: sterility, disease resistance, nutritional value, improved growth rate, or none of these. So I'd let you comment, David, on the results if you want. We see disease resistance has the highest interest for the moment.
So it seems we still need some more of you to register. You come in, which I haven't actually done myself because I've been too busy talking. So I will I will add to it myself. Disease resistance is still heading along. It's always very interesting when we compile the reports after events like this to see what the points of view and experience of the registered participants in the polls are. Disease resistance still leading the way. And about half of you have found your way to Menti by now. Clear.
Whenever you want, I can go to the next question.
We'll just give it a couple more moments. Some more people have been sneaking in, but I think it might be time to move on. You have to be quick if you wish your point of view to be registered. And I think I can be fairly confident that disease resistance is going to remain the leading highest priority trait for the audience this morning. So I think, Catherine, if we could move on to the next question.
Yes, the next question, it will be a word cloud. So what do you think are the main benefits of using gene editing in aquaculture? So you can enter a word here.
Here we go. Someone has to be first, and we're in there. And always interesting to hear what different points of view come forward. And hopefully, if all runs to time, we'll have an opportunity for some discussion at the end. And people can either ask questions or maybe clarify the points that they're coming. Some of the key points that we just had in our last presentation are obviously being referenced there. And interesting that disease came out as the most interesting trait in the last question, but here we're appearing to have sustainability and welfare implications coming up as the main benefits.
And then we can go to the next one, the last one.
I think so. We appear to have calmed down a little bit.
And of course, the results will be communicated to all of you. So the next one is, what do you think are the main drawbacks of using gene editing in aquaculture?
And somebody has to be first. So we await your, here we go. And they all come in. As Catherine mentioned, what we can do and what we will do is we produce a summary report at the end of these forums. And we found through the development of multiple of these online events now that this word cloud function gives us a very effective way of capturing some of the diverse thoughts and ideas from participants in the event. So they're a very helpful tool for us in summarizing the interest from the forums. And as I mentioned just at the wrap up to Diego's talk, effective communication is key in this area. And it's interesting what we have there: public perception, social acceptance, and public opinion being the points. As with so many aspects of aquaculture, the communication of what we do and the consideration of the social license surrounding our sector is a key area, particularly when we're considering the application of research and innovation to a fast changing sector, and this one of the fastest changing sectors of that. So fast indeed that we outpace the legislation and the policy development process, as Frank was saying at the beginning, which I found interesting, that there are complications.
Jag har helt glömt det här seminariet. Jag sitter på klimaskiten.
There's a little blast of some conversation in Norwegian.
Är det inte vanligt nu?
Understand it.
Inte ens fasen. Vad ska du ska snacka
We
här.
appear to have come.
Och vad ska jag
and
prata om?
one of the organizers could click on the mute. That would be very much appreciated. And that's very good. We appear to have calmed down with the contributions now. So thank you all for taking part in those polls. We very much appreciate your views. And as I say, we'll be ensure that we capture those responses when we write the report for it. We have to have a slight change to the agenda just for some logistics. So our next speaker is not going to be Anna, but instead it's going to be Ross Houston. Ross, unfortunately, there was another logistical problem there, but Ross has sent in a video of his presentation. So we're going to move to that now. And I think Anna's going to bring that up for us. Ross recently moved to Benchmark Genetics, which I'm sure many of you will be familiar with. His particular focus is on the latest genomic technologies towards improving aquaculture breeding. Prior to that, he was the personal chair of aquaculture genetics at the Roslin Institute, again, from whom we've just heard earlier this morning, where he led research teams focusing on genome editing technologies for a number of different aquaculture species. Ross's primary research interests include the development and application of genomic tools, as well as applying genome editing in vitro and in vivo to pinpoint functional disease resistance. He's contributed to the development of gene editing techniques in salmonids and successfully applied those to identifying genes affecting resistance to particularly viral diseases in farmed salmon. This in addition to leading a number of different projects using gene editing techniques as potential solutions for the sea lice problem within salmon aquaculture, obviously of key interest within Scotland. We have the video brought up. And
yes, and I think, David, I have the video. I'm going to launch the video. And just to let you know, because I'm not sure you are following the discussion about Anna's arrival. So for the rest of the speakers, Anna will take the last place and panelist in the agenda before the panel discussion. So we will move after to Tone-Kari and Alan, and after we will come to Anna presentations. Okay, so I will launch the video of Ross.
What I'm going to talk about is from my previous role, which was at the University of Edinburgh. Just to give a bit of context and background, domestication of aquaculture species is recent, it's ongoing, it's occurring fast, and there is huge potential for genetic improvement. You can see from the figure on the right, both the recent domestication in terms of first farming of species and the arrow denoting first breeding programs, and also if you look at the grey bar, the scale of the number of species that are required to produce a large proportion of aquaculture production compared to terrestrial livestock. As such, tailored breeding programs and genomic tools are needed. But genetics plays a key part in tackling some of the major sustainability challenges of aquaculture. That includes tackling disease and improving animal welfare, improving production efficiency, and reducing environmental impact. One of the features of aquaculture species that's very important for genetics and also for gene editing is the high fecundity. You can see some illustrations there of typical species, well-known farm species, and their approximate number of offspring that can be obtained from a single cross. This allows for flexibility in breeding program design, also high selection intensity. It allows for large-scale disease challenges, and it also provides some opportunities for scalable gene editing approaches and reproductive biotechnology. Infectious diseases present a persistent, ongoing threat to aquaculture. They're a major economic, animal welfare, and environmental concern. In some cases, vaccination, biosecurity, and other control measures are really not feasible. And as such, breeding and genome editing technologies hold a particular promise for aquaculture species. In terms of selective breeding programs, they can incrementally improve disease resistance using family selection and genomic selection. This typically requires annual disease challenge testing, and with a focus on many traits in a breeding goal, for example, in a salmon program, the rate of progress for a specific disease can be rather limited. Also, it's limited to existing genetic variation within the germplasm of the breeding program. Gene editing is likely to be a disruptive technology for aquaculture breeding. The methods are now becoming established, and disease resistance, of course, is a really key target trait with multiple environmental, animal welfare, and economic benefits. Typically, gene editing, and we'll have heard some of this today, has been performed using microinjection into early stage embryos, which has been successfully applied in a number of aquaculture species, with one of the first examples coming from the group of Anna Wargelius with the knockout of the DND gene to result in germ cell ablation sterility, and you can see also the concurrent knockout of the SLC45A2, resulting in albinism, which is used as a tracer edit. There's three main categories of approaches to using these technologies in research and also thinking downstream to application in breeding programs. One of which is to focus on QTL that exists in the breeding population to increase the frequency or fix desirable alleles existing QTL, for example, disease resistance QTL. Secondly, to look at other strains and closely related species to see if it's possible to introgress by editing desirable alleles. Thirdly, to focus on completely de novo alleles, which do not, as far as we know, exist out there. And that could be based on knowledge of the biology of the trait in question. So we take each of these categories separately. They do require quite distinct research approaches. So the first of which, looking at existing disease resistance QTL. Here it would start with a genome-wide association study, identifying regions of the genome associated with a disease resistance trait. This could include family-based QTL mapping, and then focus on functional genomics and cross-referencing differential expression, for example, between resistant and susceptible animals with QTL to try and shortlist using this sort of funnel approach from a very large list of candidates down to a smaller list of candidates, which can then be tested using gene editing in cell culture and ultimately gene editing in vivo compared to unedited wild type, for example, full sibling controls. An example of taking this approach was some research that we did in for IPNV resistance, where there's a major QTL that's previously been identified in salmon. You can see there on the left the QTL on chromosome 26. We did some functional annotation of the QTL region using whole genome sequencing approaches, and then looked at differential expression between resistant and susceptible animals, highlighting this NE1 gene as a potential functional candidate. We then took this approach of knocking out NE1 and another candidate gene CDH1 in cell culture and performed chemical inhibition 2, then challenge the edited and control cells with IPNV, harvest the cells at various time points post-infection and assess viral load either by qPCR or by infecting fresh cells with supernatant and assessing the survival. We saw that there was a marked reduction in productive IPN virus replication with the NAE1 knockout, but not with the CDH1 knockout, and also with the chemical inhibition of NAE1. And this suggests that NAE1 is the functional gene underlying the IPNV resistance QTL. So this was an interesting scientific discovery, but of course we know that for existing major QTL, mark-assisted selection can, to a large extent, achieve a similar purpose to genome editing. So another interesting approach is introgression of desirable alleles from other strains and species. There's two major consortium projects based on this premise looking at sea lice, that's CRISPR, which is a Norwegian-funded project, and GeneLice, which is a UK-funded project. And the general principle underlying some of these projects is that there's cross-species variation in sea lice resistance, for example, with coho salmon rejecting the sea lice shortly after they attach to the skin, whereas the Atlantic salmon can't. And the comparison of these resistant and susceptible species and also resistant and susceptible families within species using new genomic technologies such as single-cell sequencing and protein-protein interactions will be used to highlight genes and pathways that differ in the early stage response to lice challenge in these species, and that will lead to candidates for genome editing, potentially allowing for increased resistance in the Atlantic salmon. The third approach is really not relying on any existing genetic variation that we know about, but creating de novo alleles. This could be used, for example, to identify targets using genome-wide CRISPR screens, which have been established in cell culture in mammalian species. Here you create a guide RNA library that targets every gene in the genome. You deliver via lentivirus vector at a dose of approximately aiming for 1 integration per cell, and then you screen the cell population, for example, using a pathogen of interest, and sequence the screen cells, which could be surviving cells or it could be those with a marker of infection, and sequence for looking for enrichment of guides that inform on disease resistance or at least host pathogen interaction. And that could be adapted potentially in vivo in some aquaculture species in the future. The second part of the third approach is looking at knowledge of biology of the trait in question, and I won't go into this here. It's quite a famous example of where a host cell receptor for the PRRS virus in pigs was targeted, or one domain of the gene was targeted, one domain of the protein was targeted, and the knockout resulted in a resistant pig, which was completely resistant to PRRS virus with that small change to one gene and one protein. There's a UK-funded project at the moment looking at resistance to ISAV and using a similar approach, which we're involved in, and that's based on comparative, or in part on comparative biology of infectious salmon anemia virus compared to influenza, looking to identify genes that are tested in cell culture using a disease challenge followed by immunofluorescence, and there's been at least one interesting candidate that results in a reduction in ISAV infection. And of course, these are all interesting research avenues to identify targets, but the real key, or a real key, is then how do you then take those forward to implementation? Gene editing, of course, is a tool in the toolbox for aquaculture breeding, not in any way a replacement. It needs to be integrated into well-managed breeding programs. So the question is, how can this be done? The current methods are rather laborious, inefficient, and do result in highly mosaic animals. They're not scalable, and they're challenging in some aquaculture species. There's other potential methods which have been tried, and my colleague here, Yuaz, tried some of these methods, such as delivery via sperm-mediated transfer, or other methods of electroporation or liquefaction, likely to improve scalability in the long run. Perhaps mosaicism will remain an issue. One interesting avenue, just to finish off on, is surrogate broodstock and germ cell culture. It may be possible to isolate germ cells, for example, from broodstock animals or their siblings, culture them, then edit them in culture and transfer the edited germ cells to sterilized surrogate broodstock, which could be the same species or a different species. And this might result in more fully edited donor gametes and embryos produced from the surrogate. And this could be a way to, if the candidate siblings become the donors and surrogates become the multipliers, it could be a way to perform editing only at the multiplier stage and not in the breeding nucleus. allowing for tailored production line animals with target edits. And crucially, it could also allow for sterility to be introduced alongside disease resistance or other target traits. So that's a very interesting avenue which should be explored in the future as we look towards implementation of these technologies. And I'd just like to thank my colleagues here and collaborators and funders, in particular those named below.
It would normally fall to me now to thank the speaker. Unfortunately, I can't properly thank the speaker because it was a video clip that was sent in. But in absentia, I'd like to express our thanks to Ross for sending that video through to us. We heard beforehand how, certainly from the participants in the event this morning, disease resistance, lice resistance, sterility were issues that were flagged as being of highest relevance and importance. And I think that presentation very clearly bore that out. In addition to, certainly from my perspective, raising some interesting questions about implementation, application, and scalability of the technologies that we're discussing this morning. And that's hopefully something we'll have an opportunity to talk about in the discussion at the end of the event. But we move on now to our next talk, which is going to be given by Tone-Kari Knutsdatter, who is a senior research scientist with fish nutrition at Nofima in Norway, who's been working there for more than 20 years, looking particularly at different aspects of lipid nutrition and muscle development in fish, with the emphasis on underlying mechanisms behind that. In recent years, gene editing techniques have been included as a significant and important research tool within that. And we look forward to learning more about that now. Thank you very much for giving us your time this morning. And the floor is yours, Tone-Kari. Thank you.
Thank you so much for the introduction and to give me the opportunity to talk about sea lice resistance. So now I hope that I managed to share the screen. So is everything looking fine?
Yes.
Yeah. Very good. Yes, so today I will talk about an ongoing project on sea lice resistance. So this is like the favorite topic that you had this question about earlier. And the project is financed by the Norwegian Seafood Research Fund, where we will explore the potential of using gene editing to make salmon more resistant to sea lice. The project is led by my colleague Nick Robinson, and we are a lot of project partners throughout the world, and also 3 industry partners associated. And you can see on the right side here the logos of the different institutes that are part of the project. It's a huge project. It started in 2020, so we are just 2 years into the project period, and it will last until 2025. So, as you probably know, sea lice are one of the most pressing problems for the aquaculture salmon industry, reducing fish welfare. Sorry, it's too far there. And also causing enormous economical losses. There are several initiatives to solve the problems with the sea lice, like new production systems, chemical or thermal treatment. However, none of these methods are completely effective. But one promising fact, however, is that it's a genetic variation in the resistance towards sea lice. We know that it is a genetic variation between salmonid species. Some salmonids like coho and pink have a higher innate ability to resist sea lice infection, whereas others like Atlantic and chum salmon are more susceptible. There is also genetic variation within the farmed salmon. And this is a trait that is already included as a breeding goal by the breeding companies. But as I told, it's still not enough to reduce the problems with the sea lice considerably. But what are the mechanisms involved in sea lice resistance? So as you see here, I actually have a mistake in my title. Atlantic salmon is, of course, highly susceptible to sea lice. In the figure on the left, you see the sea louse in purple attached to a salmon skin in pink, and then proposed mechanisms involved. It's well established that certain Pacific salmon species are resistant to sea lice and are able to kill the lice within the early stages of the infection, whereas Atlantic salmon are highly susceptible. And the difference in resistance is primarily due to variation in the effectiveness of the early stage stages of the infection. Whereas Atlantic salmon are highly susceptible. And here you see the different responses in coho, pink, and Atlantic salmon, where coho will then be able to encapsulate the parasite within approximately 10 days of infection, whereas pink salmon doesn't show any of these responses. And Atlantic salmon has a huge, enormous immune gene activation. We also think that immunomodulation by the sea lice itself is also likely to play a role in resistance. In addition, semiochemical production in the fish can be involved in attraction of the lice. So then, if we manage to identify genes and mechanisms involved in sea lice resistance, then gene editing can be used to test and prove the findings. And then the potential for using the potential for creating resistance in Atlantic salmon can be evaluated. In our project called CRISPR-Assist, as Ross just mentioned, we will increase the knowledge on genes and mechanisms affecting sea lice resistance in Pacific salmon, and then utilize this knowledge to achieve full or completely resistance in Atlantic salmon. So over to the status of the project. So far, we have done a sea lice challenge test with the 4 salmonid species, 2 susceptible and 2 highly resistant. And this was done in Canada. We have selected an enormous amount of samples from the trial to be able to look into the gene expression response to the infection, performing a genome-wide association study, as well as looking into different aspects of semiochemical production. To investigate the interaction between the lice and the fish and to identify genes and mechanisms involved in resistance, highly advanced methods will be used. We will investigate if there is changing during the first days after infection, but also see if there are differences prior to infection, and then compare within and between species. The proteomic analysis is done to identify the potential immunomodulatory interactions between the lice and the host cells. So far, preliminary results show that on test samples show that we can identify a huge number of proteins belonging to either the lice and the fish, but also proteins that are shared by both. The spatial transcriptomics will link the gene expression to the position in the tissue. This method will then complement the single nuclei RNA-seq that will also be done by enabling specific measurement of genetic signals driving immune cell infiltration and response from cells situated at the attachment site of the lice. And last week, actually, we got the first successful results from this method. It's a method that goes over several days. So we are really eager to get to look into the data. You see here. Single nuclei RNA sequencing is done to understand the detailed molecular and cellular responses in the different salmon species, including the specific cellular populations that are involved. And so far, we have managed to sequence a good number of genes per nuclei, and then also identified marker genes for several cell populations. Semiochemicals or chiromones are thought to be involved in sea lice attraction. Our chosen candidate genes are involved in that are involved in semiochemical production belongs to the cytochrome P450 superfamily. And we have selected some that are only expressed in salmonids. And we have had a master student that have been looking into the gene expression of some candidate genes belonging to this, this family. And she has actually found some very interesting results. Some genes that are highly expressed or only expressed in salmon and not in the other salmonid species. And then we will also look into the different semiochemicals. So it's a huge job just extracting these semiochemicals from the water of the challenge test. But it's that we are in the process of this part of the project, and we have run some test samples showing a good profile. And then later on, the knowledge from this from the characterization of the semiochemicals will be used to do a sea lice behavioral test. And then to see what kind of products or compounds attract the salmon, and also if there are any different parts of the salmon that attracts the tissue samples that will attract the lice. And then in the end, when we have all the results from the different methods, a meta-analysis will be done to select the candidate genes for to be used in a gene editing study. But the strategy for what we will do for doing this, or the strategy for gene editing, will be decided upon when we know the different genes. And ethical, social, and legal implications of using gene editing will be evaluated through a responsible research and innovation approach. An important part of the project is also to risk assess and model the potential for the sea lice to evolve to overcome the genetic resistance in fish. A model is developed for one gene, but when we get more knowledge on the different other genes that might be involved in resistance, this model will be updated. So at the end of the project, we hope that we have identified genes and mechanisms that are involved in sea lice resistance, and that this can be used as a basis for new strategies for to deal with lice resistance in Atlantic salmon, either using gene editing or breeding or a combination. And in this way, we hope to support industry with a solution to tackle the problem with the sea lice. Then I will just, thanks for the attention, and then also thanks to all the partners of the project.
Thanks, Kari. Thank you very much for this very interesting presentation, going more into the details of the sea lice resistance and the project in which many companies and institutes are involved. We are a bit late, so I'm going to introduce Alan very quickly. So Alan Tinch is now working from 1 year now, I think 1.5 years perhaps, at the Center of Aquaculture Technologies. Previously, he was working in benchmark genetics, and he was a former member of the FABRETP board representing aquaculture. During his career, he has introduced new technology into breeding programs to improve health, welfare, and performance. And he will explain us now how the Center of Aquaculture Technology is exploring commercializing options for genome editing in farm aquatic species. Alan, the floor is yours. Thanks.
Good morning. I'm speaking to you today from San Diego in California, where Kat is based. The organization of this event and the trip that I had to make out to California has coincided, so I'm speaking to you now at about 2:30 in the morning, I hope I manage to remain awake for the next half hour, but I'm sure if I fall asleep at the wheel, somebody will nudge me and let me know. I want to talk quickly about when and why we'll see gene editing in aquaculture, particularly from the perspective of Centre for Aquaculture Technologies, where we're commercializing this technology for use in aquaculture. Very quickly, in probably less than 8 minutes. CAT is a commercial organization with expertise in gene editing and genetic technologies. It also works in feed and nutrition and health research supporting the aquaculture industry. Our operation in genomics and gene editing is based in San Diego, California, and our nutrition and health labs are in Canada. We are bringing technology to the industry, aiming to have a number of different technologies available under one roof to support development of new technology in aquaculture. In terms of where CAT sees the technology, a number of statements looking at the technology, we regard gene editing to be possible in all of major aquaculture species. We think legislation will become more permissive in an increasing number of markets. We intend to work globally with breeders and producers to bring that technology to the market. We see, as a number of the other speakers have identified in their talks, that sterility is a prerequisite to effective use of the technology. The changes that we make are to the fundamental structure of the DNA, and we want to see that under control of farmers. Not all fish farming is possible under controlled systems such as RAS, and so we see sterility as being an important aspect of how we bring the technology to market. The route to market is through established breeders and producers, and they already deal quite closely with the needs and expectations of producers, farmers of fish, and the consumers. So we need to understand each other's concerns and apply the technology in a constructive way. And our intention is to use gene editing to bring sustainable benefits to health, welfare, and efficiency. And I echo the respondents to the surveys earlier on that with the important traits that are the important aspects of the technology that will continue to be important to us. We see gene editing as the next revolution in breeding. Why do we want to bring in gene editing? Well, essentially because not all the traits that we deal with as breeders and geneticists are able to be changed by conventional methods. There are traits that we describe as being more difficult than others, and many of those traits are the target traits that we're aiming for gene editing to be applied to. So it's things like disease resistance, where the genetics is difficult to assess and difficult to manage using conventional genetics. We see potential solutions using gene editing. We want to work with the sex of the animals. Often in a farming scenario, we only need males or females. This can be seen in most extreme in terms of dairy cattle and laying hens, where you only need the females for production. So there are real world examples where management of the sex of a population is very important. The same happens in fish farming in many cases, where it's highly desirable to have one of the sexes, male or female, which has particular performance characteristics that are desirable at a farming level. And we can get welfare issues developing where one of the sexes may mature in the production system, and that's very undesirable in terms of production and welfare aspects to the animal. We want to work with fertility, particularly because sometimes you don't want our animals to reproduce, particularly in the production environment, the final environment. We don't want animals to coming into reproductive condition, and so we need to manage the fertility of the stock. And there is the issue of when we're farming in normal conventional production environments, there is always the chance of animals that we've farmed escaping into the wild, and we want to minimize the effect on natural populations and prevent interbreeding of the farmed animals with the wild stocks. And disease resistance is is rightly identified as one of the major target traits, and we see that it's right and proper for us to address the issues of disease. And using gene editing, we have a new tool to target genetics that affect disease resistance and give natural selection a boost. And then there's the economic aspect. It's from a sustainability point of view, it's important that we're making animals more efficient in terms of their growth, so faster growth, less feed consumption, and increasing the yield of the product. So all of those affect the economic sustainability of farming. In cat, the company Center for Aquaculture Technologies, we have in finfish already gene editing operating for infertility. We're able to manage the sex of the species that we're working in, so we can produce males only or females only, and we're working on functional traits that are delivering better performance in the animals that we've edited. However, it's a scalability issue. We need to be able to move the technology from the lab into production systems, and in that structure, we have to see gene editing in context. All breeding programs work in a system where there is challenge from pathogens, and it's fundamental for all breeding programs to work in a high biosecurity environment. And then there are a number of fundamental technologies that we need to build in to our breeding programs, gene editing, genome editing being one of those. So our view is that generally genome editing will not be applied as a tool on its own. It will be applied in alongside other established technologies. So typically, breeding programs that are already in operation, that are well organized, that have the structure and an organization to deliver genetics to customers will be those companies and organizations that are capable of bringing in genome editing. So it's important, I think, to think in context and not regard genome editing as the only tool in the toolbox. It comes alongside a large number of other technologies that are already well established and common in the breeding programs and organizations that are supplying our fish and other livestock into the farming structure. We deal a little bit with a paradox, the infertility paradox. How can you breed for infertility? The animal breeding programs rely on genetics and breeding to improve the performance of the animals in their systems and improve and multiply the improved genotypes into the farming programs. Business depends on those improved genotypes being able to be bred in large numbers. But if the genotypes are infertile, if we're breeding for infertility, then the program ends and we don't have any fish for the farmer. So we have to be able to breed it in order to farm it. So we have to be able to separate the control of infertility from the production and multiplication. So we're working on genetic and reproductive technologies that aim to deliver the desired genotypes. So looking at conventional genetics combined with edits for functional traits, delivering the desired phenotypes, the ability to reproduce those genotypes in an infertile state, and then produce infertile production fish from the gene edited lines. Already we have technologies that we're ready to deliver in these areas. We see those in a number of in three main areas. On the left of this diagram, you can see what's a schematic of a conventional breeding program in fish with a breeding program running using conventional technologies at the top of the pyramid, producing multiplier parents who then produce commercial eggs or juveniles. We see gene editing fitting into that program as providing the commercial, the new genotypes that are aimed for commercial production. So that may involve gene editing for sterility and single sex. It may just be a high value genetic stock, and we're using germ cell extraction in that process to bring to get control over the genotypes that we want to multiply. We see there being a need for surrogate broodstock, where those will be carriers of the genotypes that we want to bring to market. And then we combine those two processes together through a process of multiplication and germ cell transfer to deliver the commercial eggs and the commercial juveniles. So where and when? Where the legislation is favorable and where consumer demand exists, and where we're able to identify those genes and the variation that we want. So typically in the areas of health, welfare, and performance, we can bring gene editing into that structure and use it as a means of developing the genotypes we want. We see sterility of those edited strains as being a critical step for the implementation, for the control of the use of the technology within the production environment. And that's a fundamental prerequisite of the work that we're doing. We recognize regional concerns about gene editing. Europe has, over the last few decades, developed a principle, the use of precautionary principle, looking closely at the developments of new technologies, particularly GMO, being opposed by green politicians and operating within the environment. There are more permissive environments that we see around the world, particularly USA and Asia, where there is more reliance on the science and acceptance that the development of the technology may be used for good rather than just be a problem and applied in a negative way. And then we have discussions with colleagues typically in the Southern Hemisphere that are looking more at an approach that says, show me how this works and we'll see how we can develop it within the system of application. The role of the breeder is to offer products that are produced using gene editing, help assess the results of the performance in those animals, and then provide that option for the consumer, and the consumer should be informed enough to be able to make a choice. And from a commercial perspective, CAT are building the technology just now and are ready to apply it into breeding programs as we take the discussion forward. So thank you for your attention. It's a short talk. I think I managed to stay awake for most of it, and I look forward to any questions that you may have in the panel session.
Yes, thanks, Alan. Thank you very much. You were not late. We are late, but it's not because of I think. David.
Yes, I was going to say, Alan, thank you so much. Not only for managing to give us that talk at 2:30 in the morning. I hope you're able to start your official working day having had some sleep, but to start talking to us about the existential issues of the infertility paradox and explaining it clearly at 2:30 in the morning deserves a special mention. The infertility paradox is not something that one routinely talks about at that time in the morning. We have to move on.
It's not the first time I've talked about the infertility paradox at this time of the morning, but there you go.
Commendable action. We have to move on quickly. Hopefully we'll have a chance for a quick chat at the end, but our next speaker was listed earlier on the agenda. Anna, who is joining us from the Institute of Marine Research. Her presentation is just coming up. Anna holds a PhD from the Institute of Molecular Biology in the University of Bergen in Norway, and is well published, numerous scientific papers in the field of animal genetics. And Anna will be talking to us on a prospective and potential uses of gene editing for salmon breeding and production. So Anna, over to you. Thank you very much.
Thank you. Do you hear me?
Yes, very well. Thank you.
First, I just would like to say I'm sorry I'm late. There was something with my calendar this day. It was listed the wrong day in my calendar, so sorry. But I have a presentation that I'm going to talk about the work we've been doing on Atlantic salmon on gene editing. And we've been working for many, many years with gene editing and Atlantic salmon. And we have several lines of different mutants that we are currently having. And I'll come back to talking, just give some examples today, because I've seen that a lot of people already gave a lot of background. So I'm not going to focus on the background, just give 2 examples of fish that we have currently that we're doing science on. So, and also I want to go back and say, it's not only like the use of gene editing in aquaculture, also it's very, very important to understand the biology of the fish. So there's like 2 perspectives, because this method is of course extremely potential to try, trying to understand, for example, important life cycle stages in fish and aquaculture. For example, salmon, we work now with multiplication, which is transition to see what, just to understand that, or just to understand how basically puberty is controlled. And then you don't even have to use the gene edited fish. It's more like so much more of biology, so you can adjust the protocols of breeding and also rearing of the fish. So this is showing a picture of our facility in Matre, north of Bergen, where we don't currently have any gene edited fish because it's in the sea, but we have all our fish done in indoor facilities in Matre. And the picture is showing a simple edit that we did. The first trying out is knocking out a gene encoding a gene silhouette carrier for DFI-A2, which encodes genes involved in pigmentation of salmon. So I've seen so nice presentations earlier today, both about disease and also haven't seen it about feed, but it's also an important area. So major sustainability challenge in Atlantic salmon aquaculture is, of course, what you saw before with Karianne showing, and also Ross showing about the salmon lice. The feed is a big issue now because we have to have locally produced feed, and the feed has to be then adjusted to the fish. So also you can adjust the genetics of the fish so the fish can digest the feed that you're interested in. But then what we've been working with a lot in my group is then the problem with genetic integration, and into that area that Alan just talked about, the sterility. And also we work a lot then with welfare traits, because the main task of our institute is then to give advice to the government about sustainable solutions, but also about welfare of the fish. So in my group, we've been working with gene editing since 2013. And we have been working with a number of traits, and we have used both knockout technology, which is the most simple technology. When you CRISPR-Cas, you just cut the DNA and you get the cell forms a mutation. And in this case, we work sterility, maturation, pigmentation, omega-3 metabolism, multiplication, disease resistance. But we also then work with more specific methods where we try to make SNP exchanges, and inserting whole genes, and where we also worked in a number of traits. And it all started then in 2013, where we started to do our first experiment, and this time we were only injecting into first cell stage embryos, and then what we could see if we CRISPR the fish then with the gene encoding this pigmentation gene, as we could get a full mutation of that gene. Both alleles were targeted, so because you can only have this phenotype if you lose the both alleles. So showing that CRISPR is very, very potent, very efficient in salmon. But when you look in more closely, like we've been doing now, we're working on several generations, we see that it's often a little bit difficult in the first generation. This is broodstock fish, or one of the mutation types we've been working with. And as you can see, you have a number of different mutation types in the fish. And also another thing that we are going in working with now is seeing whether the CRISPR is mutating somewhere elsewhere. So it's both what they call off-target and also then the mutation types. So what we do now, we've been working a lot with the F0 because of the generation time in salmon, which is very long. For females, it's 3 years, but for males, we can come down to 1.5 years efficiently, but still a fish that take very long time to get to the homozygous mutation that we want. But what we're doing now is that the mutations we created 3, 4, 5 years ago, we're now crossed out to make F1 generations illustrated here. So in the first generation, you have many mutation types, and these can also be not only knockout phenotypes, it's also you can get sort of in-frame and gain of function mutations if you're unlucky. So the F0 is interesting, but the F1 is much more interesting. if you say so. And I'm not going to show you this, but then I'm just going to give an example of sterility that we've been working with, the genetic integration. Here we've been working with known genes from mammalian and studies where they know that these genes are important for germ cell formation, to see whether we can target any of these genes to make the fish sterile. And why we've been doing this is because the methodology so far used for sterility in farm fish is triploids, and triploids might be useful for some species, but in salmon they are not very useful because it causes a lot of welfare problem. They get very sensitive to environmental changes. You have increased prevalence of skeletal deformities, and also the males go through puberty, so you have the welfare aspect on that too. And they seem to be more sensitive to bacterial skin disease, and they're not always 100% sterile, so it's not the perfect case if you have an edited organism. So what we did a couple of years ago, we had a project where we knocked out genes involved in germ cell formation, and one of them was the dead gene. I think this is already presented by other here today, but we knocked out this gene, and what we could see that we didn't get any formation of germ cells. This is then showing a female control where you have the oogonia, but the knockout female don't have any germ cells. And this phenotype is interesting. In many cases, not only for sterility, but also for surrogate productions. We currently have a project where we're doing surrogacy of these fish, coming back to what Alan presented just. But also these fish are also interesting because they don't have the welfare problems by going through puberty, so they don't enter puberty. And recently, we're soon going to publish a study where we look at the welfare of these fish, and it seems like they have a very nice welfare, these fish. They're not distinguishable from wild fish, from other farm fish, unless that they don't have this final growth part because they lack the sex steroid. So it might be you have to maybe tune to get a better final growth of the fish. But the problem to breed this in masses is, of course, that you don't have any germ cells. So for a couple of years now, we've been working with the project where we are rescuing the germ cells in these knockout backgrounds. So we knock out the dead end gene, and then at the same time we add the dead end mRNA that encodes the dead end gene. And what we have seen is that we can get germ cells both in males and females. And we also now cross them out and see that we have sterile fish in the offspring. And we have these fish that we have rescued them to make new germ cells again, but we haven't analyzed those. So just showing you how such a fish look like. This is then a fish which is a dead and knockout female, and you see you don't have any oogonia. This is then a fully knocked out dead end fish in the F0 generation, and you have oogonia in this fish. And we have now worked a lot with the broodstock of this fish, and it's a little bit frustrating because the females we seem to be able to get the very high efficiency of the mutations, but the males, maybe the sperm is not as efficient in keeping the mutation. But so now we're just working on the F0 generation. So looking now, working with the F1 generation in the coming years will be very interesting. I also wanted to give you another example, which is the welfare. One of the welfare parameters that we were working with is early puberty, which is then a male problem in the farms. And these fish then get increased. This is susceptibility. They stop growing and also have osmoregulatory problems. So it's not very nice to have them. And this happens. Now they're controlled by using continuous light in the farming facilities, or also genetics like the late maturation allele. However, with increased use then of RAS facility, online facility, and at higher temperatures with climate change, we still have this problem with early maturation, which is then a large welfare problem. So we are working with the gene what I call the forever young gene here, which we are now crossed out to second generation. And here we can see fish that are then knocked out for this gene. And we had this fish in a common garden together with wild type fish, where we have them stimulated the fish to enter maturation. And what happens there, that's all the control fish, they do enter maturation, but this, what I call forever young fish, they don't enter maturation, they stay at the spermatogonia A stage. So this is another method then to have higher welfare of the fish. And also one of the interesting features of this is that when you look at the heterozygous trait or the intermediate trait, you see that they also have less probability to enter maturation. So that you can also, you don't have to always use a knockout trait, you can actually use the heterozygous trait. So that's another thing what comes into when you do breeding, that some traits are coming as good as if they are half the genetic addition. So just wanted to give you some examples of the work we're doing, and in the end I want to acknowledge then my group here at the Institute of Marine Research, the Population Genetics Group, and our collaborating partners, then our University of Bergen, Uppsala University, and NTNU, and University of Utrecht. And thank you for the attention.
Thank you very much, Anna. Sorry for the confusion. I mean, perhaps we were not clear about the date from our side too. We are running very late, and we have seen that there are some questions in the Q&A box, but that have been answered. So we wonder if you have a last question, and I wanted to know, Frank, you raised your hand before. I don't know if you had a question for one of the speakers. I think it was Diego. Wanted to know if you had the panelists, you had a question from someone else in the panel, and in that case we can give you the floor, and after we can I can give the floor to David to summarize and to ask you for a final statement to all of you. Frank.
No, it was just an error on my side. I clicked the wrong thing and said, what have I done now? So, but it's fine with
okay.
me. Thank you.
You have any questions to the rest of the speakers, the scientists?
No, I'm baffled by the information.
Okay, David. Well, just let me check if there is a last question in the Q&A box. Very interesting questions, and but they were all answered. We will provide them with the report. David.
Thank you very much, Anna. It's always the way at the beginning of an event, you think you're going to have all of this time at the end to have a careful discussion and provide a detailed and nuanced summary. And unfortunately, it never quite goes like that. I said right at the beginning that gene editing can sometimes be thought of as being a quite specific and technical subject area. But as we've heard, it is indeed far-reaching, addressing every aspect of our sector. And I think that's very much been borne out this morning. Some things that have struck us have been how fast-moving this area is. And whilst fast-moving enough to prove a challenge to the legislative and regulatory process and the quite formalized process that goes through, I think we're all, we must remember and be very reassured that there are safeguards concerning impacts on human, animal, and environmental areas. And the principles and ethics are something that are not at all sacrificed within a wider context. Although it was interesting to note there are differing global perspectives from the way we approach the subject matter in the EU compared to, as we were just hearing laterally there, within the USA and Asia, and also within the Southern Hemisphere. One thing European technology platforms do is that we comment on research and innovation research priority areas. And certainly within the Horizon Europe framework and other areas, I think they need to invest in policy actions on animal research, and the public perceptions relating to that is something that Anna and I will take away in our work and consider how best we can continue to develop that. When Diego was talking, I was reminded of Japanese haiku that somebody sent me once, which states, "Evolution is a work in progress. Are you taking part?" And I think that's quite a nice way to approach some of the issues we've addressed. Gene editing, gene variation occurs naturally. And what we are doing is looking at modifying that and using that tool. There are clear benefits, as we've heard, in terms of feeding the world, environmental impacts, aquatic animal, in this case, welfare, and improving health, the reduction in reliance on antimicrobials and so forth. But then there are issues relating to that in terms of the application, implementation, and scalability on it. And that remains a key research and innovation challenge in so many areas of our work, how we upscale and implement some of the technical capabilities that we have. It's also quite clear that gene editing remains a tool within the aquaculture sector. It's not presented in any way as the only answer. It's part of a toolbox approach within farming. But the benefits of that we've seen, be it relating to sterility, fertility, and sex, be it relating to issues such as disease, the economics within farming, and as a tool within the context of approaching lice and other parasitic issues. As Anna's mentioned, a report comes out at the end of these results. The event has been recorded, and so we will pore over that and consider some of the nuances of the presentations. Questions have been asked within the Q&A that have been answered. And if there are any subsequent issues that we think require clarification, we'll come back to our panelists. You're all warned on that we might come to you with a quick follow-up just to clarify our report, and we'll share that with you. I know that EFFAB and FABRETP have their AGM next week, so I'm sure, I know that for a fact, and I'm sure there will be much communication coming out of FABRE and EFFAB in relation to that. The next EATiP event is an On the Horizon project dissemination service event, online event, where we present outputs from various EU-funded research projects within environmental impact. That takes place on the 12th of May. Many thanks for registering and participating in this event this morning. And be sure to follow our respective organisations to make sure you're kept up to date with what other events we're organising and running in the near future. So a particular thanks to all our panelists, wherever you are in the world and whatever time it is you spoke to.
Yeah, especially to Alan.
Especially to Ross in absentia, who unfortunately couldn't join us, but did send a clip in. And my own personal thanks again, Anna.
Thank you very much. I thought we were going to ask for final words to our speakers. No, David, we don't have time to ask the speakers. They would like to have a last word, a final statement. I think we had time for this. I'm sorry, I misunderstood what was in our private chat. But don't worry, it's good. I mean, it's 12. So thank you very much, all of you, for coming. We keep in touch. All, Frank first, perhaps. We will see very soon again. And thank you very much, all of you. We will, bye-bye. It's always difficult to say bye-bye in a webinar because we have gone back to the in-person meeting. So we would like to take a coffee or a drink with you now and to exchange more. But yes, it was nice to see you, and see you soon. Thank you very much. I'll attend this too. Bye-bye.
Thank you all. Nice to see you all. Bye-bye.